looks like the top of a Phillips head screw or or a Nautilus symbol.
Our compass rose.
Yeah, compass rose, yes, compass rose, not Nautilus.
Nautilus has rights to the compass.
I don't think that's true. Video pushing.
Like this at Rorschach's test.
Yeah.
A lot of the maps are.
Yep.
The ones that are posted on the walls, I don't see a lot of the things that are listed as being seen.
Lindsay's sunglasses.
Lindsay's broken glasses.
I see that one.
I like that.
Oh.
Looks like another type of corallium or hemicorallium.
Shrimp and then
Oh, current.
brittle stars. I mean, you feel like you almost
All
never
right,
see
I'm gonna
like,
get
these
lined back
corals
up in
without
front
like
of Jess.
associated brittle stars on them.
Yeah.
It's pretty.
Thanks, Marley.
And again, like we saw those really young looking ones, those really small recruits, and they even had brittle stars.
Yeah.
So it's
Yeah.
indicative of
Do
that.
the corals
I don't want to
like-
end
like
up
the
in
brittle
off my
stars?
rhubarb.
They don't seem to be too upset about it, right?
But is there any evidence that they're actually beneficial?
There is from a disturbance standpoint. So when the Deepwater Horizon happened in the Gulf of Mexico, there was impacted coral colonies. So corals that had what we were calling flock, so like oil and dispersant material actually on the coral. And the corals that had brittle star associates were covered at essentially a faster rate than the other coral, any coral that didn't have an associate. And that was done through like image analysis. So scientists went back every year, took the same photo from the same angle and used.
Yeah, I think Alvin was part of that. Little Herc was part of that. Big Herc was part of that. Jason was also part of that.
Yeah, there was a ton. I mean, Oceaneering was also like the Global Explorer, as well as a couple other vehicles.
Surprised there's no current like right here.
Oh, really? Wow.
That's a good point.
Is current something that are you mostly sensing that visually, or are there sensors, or is there a way you can kind of feel
There's
that as you're
no
driving?
real sensors. It's
Your
if I know
power
how my Z bias, my vertical bias is dialed in, then if I'm hands off, like what's happening. There's also just watching the particulate in the water column.
Mm-hmm.
I wonder if we were on the other side of this ridge, if it would be a different story.
That's what you were feeling just a second ago, wasn't that?
Yeah, there's current on the other side. But it was pushing me west.
Oh.
Kind of going around a pretty vertical looking cliff face here. There's quite a few corals still on it.
Yeah, the vertical is probably a pretty popular spot for the corals.
This is kind of it looks to me at least like a much more rubblier terrain than we've seen before.
Yeah.
Is that indicative of any type of formation here? I mean,
Well, I think the real question is it a scree slope, Megan?
Oh dear.
Oh, look.
It's definitely not.
Video, let's push in. Keep going.
Small little Anthomastus mushroom coral.
Yep.
And a new little recruit there at the base. See that separate little one polyp?
Oh yeah.
That's how they start. So they
Really?
start as one little polyp, and then it'll just continue to bud polyps as it grows.
So that type of coral are they reproducing asexually then, just sending off little budded polyps, or?
I think they can do both. Yeah, so I think they're doing both. At some point, it will probably reproduce sexually also.
Okay.
It's an interesting gall on that coral right in the center of the screen.
Oh yeah.
Sometimes coral tissue will grow over something that's like settled on it.
Mm-hmm.
So that could be that. It's a beautiful image.
It is a really beautiful image.
All right, video, let's frame it up.
It's interesting that the corals will form those gulls as well,
Mm-hmm.
because that's terrestrial, like plants
Ah,
will do
yeah.
that as well.
It's very much a tree thing.
Yeah. It is a huge
Brittle star.
Is that
Yeah.
all one brittle star?
It looks like
Yep.
it.
Wow.
Oh, wow. Yeah.
All right, Marley, let's come full line. Let me square up on my... All right, we can bump in.
Yeah, so typically we've been on less steep slopes overall while diving, so we haven't seen very many rubble shoots or pillow talus, so talus like broken up.
All right, Marley,
Rock.
we're pushing in again.
Yeah, even with the smoothing of gridding for this map, it's like 40 meters. There's a 100
It's
meter
another one of
rise.
these ophirites.
Yeah,
Wow.
it's
Brittle
really
stars that's
impressive.
just on the rock, but
That is not a healthy coral.
no. So most of that looks like that hemicralium is dead. Just the little bits of pink polyps are what's still alive, and then the yellow is zoanthid, so that type of coral that will colonize the skeleton of other corals.
Oh, really?
Yeah, so those polyps are, I wouldn't say parasitic because we don't really quite know the relationship, but they'll find corals that are either dying or
There's one of those
dead and
sponges.
just start growing.
They only associate on the exposed skeleton. They're not like inducing.
So
the
that's
death.
one of the things that's not quite
Really?
understood. Yeah, because sometimes you do find them growing on what looks like a
Over.
relatively live
Right.
colony. So I don't know for sure. That brittle star is all tangled up down there too. Yeah, and that's some of that sponge where they have
Yeah.
those more like spine-like projections.
Yeah, it's like a crystal.
You've got-
Yeah, it does.
I like seeing the hydraulics of the leg unfurl on that brittle star.
Look at those arms.
Are those arms part of their foot? Are those projectile?
Holly, is your mic
I don't know
on?
for sure. Oh, yeah.
Okay, Marley, full line. Oh, that's a big crazy sponge.
So after a couple more steps, I may bring us due north.
Okay.
Does that sound good to you?
Sure.
Yeah.
Jess, I'm sure you've noticed this, but I tend to fly with a vehicle heavy, like on Z-bias.
So you don't float away from your image.
That way, yeah, basically I know that I can just, if I'm flying in towards something,
Can we take a zoom on this one real quick?
as I'll just kind of like settle down
Looks like
slowly
that.
onto the rock.
Yeah.
It makes for nice, easy landings. And usually I don't have to then drive myself down at any point. And I'm really just canceling the drive down to come up.
That's
Short
fine.
video, let's go ahead and push in. So I'll keep, Ed says I can be a pain because I'll keep driving forward while after I call for the zoom.
This
trying
does
to
definitely look like
Keep
some
stuff in the frame.
more oh, there's a cup coral there too.
Oh, wow.
More Paragorgia-like, some more like the bubblegum corals.
And cup coral.
There's a cup coral down below. Oh, I see.
Yeah, there's a couple of them. Little solitary corals. Most corals are made up of a colony of polyps, essentially. So each polyp is an animal, and they all work together for the whole organism. And the cup corals are just one polyp, so they don't have that colonial organization. So just
Aw,
hang out on their own.
just by themselves.
Loners.
How do they work together?
So each polyp is a mouth, and then so they're all feeding and they're all contributing to essentially like the overall health of the colony.
Yeah, we're getting close
All
to
right,
the
full
waypoint.
wide.
Are there any corals that have many polyps where the entire coral is a single organism, even though there are many polyps, or anytime you see multiple polyps, each polyp is a different animal?
Yeah, anytime you see different polyps, each
They're all
polyp is,
different
yeah.
animal,
We didn't really comment too much about it, but the basalt that the cup coral was on just now was really cool. Like a really
Yeah.
sort of a bubbly
Yeah, it was really
texture.
Botryoidal.
What was that?
Megan, you could have a convert. Play your cards right.
Yeah.
So the texture was it looked pretty botryoidal. We're seeing that on a smaller scale as well. It's kind of like the manganese crust that's growing on the surface of the basalts. Instead of just growing kind of as a smooth, flat surface, little sort of
Video, let's
disturbances
push
or
in.
sediment, it's going to grow around. So if you kind of picture the
Hold that.
growth sort of going outward and round next to many other
Alexis, are you
places
ready
like
for us
that,
to start
it's
heading
going to have a lot of
to
sort
west,
of
to the
bubbles.
point?
Yeah, I
Okay,
think
cool.
so.
Great.
And over time it'll grow bigger.
Heading west.
There are other
Yeah, I'm going to put in two...
minerals that have similar habit,
put in
like
270.
malachite. I know it does this
a little more than our bearing.
Are you heading to waypoint 2?
Yeah.
It's kind of like really primitive nodules.
Help pull us back in line a little bit.
Yeah,
Cool.
I probably should have done it a little earlier, but I was looking at
Sponge
this route.
Yeah.
plus a coral. Oh,
Is
looks
it a
like,
whip coral?
yeah, looks like maybe a black coral.
Video, let's push in all the way. Yep, black coral.
Yep.
It's amazing this specialist is the winner. All right. Full wide, Marley. Thank you. Josh doesn't like us flying at 100%, and so I've been very conscious in the past year about setting a joy gain, because I've never used joy gain before.
Oh no.
So you can fly at 100% at 50%
Yeah,
joystick?
basically.
Some smart thinking there. So when you're doing ascent and descent, are you just setting a Z bias and letting it do? I see. Okay.
Yeah. Some people still stick lock. I don't know why.
No.
I'll stick lock forward, but you know, if you can control one, like control them separately, there's no reason not to.
Yeah.
What is that?
Dead sponge.
Weird rock.
Rocks are pretty tricky.
That's one thing we've learned this last
Well,
couple of dives. Rocks are
anemone.
tricky.
They will trick you.
Alexis, what are some of the things that we're looking for on this dive? Like when you're looking around and trying to decide what you want to collect?
We try and collect just a like wide
time around,
representation of
270.
what the community looks like. So, and then we
I'd wait
have
until you're
a
closer,
number of
gets closer
studies
to
that
the
are going
stern of
on
the,
that
like,
have to do
north-south
with
even with the
the
ship.
overall essential Pacific seamount distributions, and so we're trying to collect anything that looks incredibly different from something we've seen before, because it means we wouldn't have genetic material for it yet. And then anything that looks really representative of like this specific seamount. So like with the hemicoralliums, we're pretty confident in what that is, so it's not as necessary to take a physical sample as long as we have some good imagery of them. But then, like earlier, right before we came on watch, they sampled what looked like some type of Iridogorgia, but it had branching that was not normal, so they sampled that because it's something that we think we might know what it is, but we're not positive.
Right.
So that's where the genetics really helps, and the morphology, having a piece of it to look at like the actual polyp orientation.
Okay.
So this is a good example, Ben, of like what you asked about with the zoanthids, because this coral is still like very much alive,
Right.
but it already has zoanthids colonizing it.
So if you were to take this back to the lab, if you looked under the hydroid polyps, would you likely see soft tissue still of the?
That's a good question, and
Because it's like, so.
I think it depends on when they colonized. I think, yes, the newly colonized material, so like at the ends of where those zoanthid polyps are growing, maybe there's still soft tissue under there, but I think over time that soft tissue of the pink coral would be suffocated, essentially.
Yeah, I was unsure because I usually have seen them on the exposed skeleton, so I didn't know if they can recruit to the soft tissue, and it seems like induced coral death themselves.
Makes for a pretty picture, the pink and yellow, but not necessarily. I feel like it looks like Starburst to me. Like this is like somebody squished together.
Delicious.
Yeah, sure does.
Rich.
Smashed up a pink and yellow
Very
Starburst.
strawberry banana.
Yeah, yeah.
All right, video, let's come on.
I think the slime stars look like disease starbursts.
That's true. Like a radioactive starburst.
Yeah, yeah.
That sounds good.
Add that to the food blog, Ben.
Cool sponge back
That is a
there.
cool sponge.
It's like a
Smell the
balloon.
umami from
It's
here.
very bulbous.
Yeah. Wow, it's a really cool like internal structure too.
Yeah, most sponges have like crazy canals inside.
Oh,
And they're so interesting that you see them like on the seafloor and you feel like you would understand the texture, and then when you bring it to the surface, it's much different. They're
Really?
really like, yeah, those types of sponges are like really tough and crunchy.
huh.
Yeah. The glass sponges, because they're made up of silica, so it's
Right.
essentially like a bunch of tiny glass shards.
Oh wow.
All right, starting to do our heading change.
Oh, there's
It's a little stock crinoid.
pretty, yeah,
Five
stocked crino.
5 arms.
And 5 arms because it's an echinoderm, right? Are all echinoderms 5 arms?
No.
Or 5
They have radial
5
symmetry.
radial symmetry.
Yeah,
Right.
but not all of them have 5 arms. But those 5-armed echinoderms are, I think, kind of unusual to see. The ones that have just 5 arms.
Really?
Yeah. Or crinoids? Did I say echinoderms?
You did.
I meant
yeah.
crinoids.
Crinoids.
We understand.
You guys get me.
Yeah, the cleavage on those bigger boulders look totally different than the smaller stuff.
Do you know if they found a manganese nodule?
Did we find a manganese nodule, Megan?
No, not an official, like, sort of solitary nodule in that sense, but we've seen a lot of botryoidal textures on some of the basalts we've been pulling up. Some of them are really significant, look like little manganese nodules.
We'll take a look at the little primnoid down there.
Video pushing.
Hello, primnoid coral. Oh, it's got something. Is that an
looks
egg sac on it?
No, it might be a medusa. Sometimes little like medusa jellyfish stick on them.
Yeah, I don't think it's an egg sac.
Yeah.
That up near the top on the left.
Yeah.
Yeah.
Yeah, so we've seen it before, only a couple times. It's like a tiny little jellyfish. It looks like it's stuck on there. Steve says this is Calyptrophora, but the colony is too small to determine species from branching pattern.
All right, Marley, let's come full wide.
Yeah, one of the great things about being on Nautilus is that it's not just the scientists that are in the van right here, but you guys have groups of scientists that are watching live and chatting in to assist with identifications or with their own research as well.
Yeah.
And just because it's fun.
Yeah. Yeah, there's a lot of people that just turn it on like a second screen at work, and you're working on other stuff, but keeping an eye for if there's anything that really is something you want to comment on or contribute to, which is really beneficial. Like some of the benefits of telepresence is that you just don't always have to be on the ship. For a lot of us, you know, we like to come to sea, but you can't be on every expedition, so this gives you the chance to participate at least somewhat.
Yeah.
Right, and we've had chief scientists who were not on the ship before, you know, run the dive.
Yeah.
Oh, wow.
Decide what we're going to sample and whatnot.
Well, we often have Nautilus Live on our second screens in my office in the U.S. Fish and Wildlife Service building in Honolulu, so...
Yeah, my next cruise will be the chief science is like 8 months pregnant.
Oh.
She's going to sit in her space center.
Cool.
Oh, that's awesome.
Yeah, that is awesome.
Yeah, one of the other benefits, so these dives are so beneficial to all sorts of researchers, but it's also beneficial to us. So for the U.S. Fish and Wildlife Service and NOAA, who rely on this type of mapping and data to better manage these spaces that we still don't know a lot about. Ooh, and we got a fishy.
Yeah, fish.
All right, Marley. Let's get a fish.
Going fishing.
Let's get a fish.
Oh, is this our first fish of our dive watch?
At least zooming fish, yeah.
Ooh.
It's got a big old head.
It really does.
Doesn't look like he has very pronounced eyes, does he? I guess this is
Oh,
further
coming
forward.
around.
Might be a type of snail fish.
Looks like a snail fish.
Right, okay.
He's ran away. Nope, he's in my shadow.
And so we started this dive at, what time? 6:30 local.
Yeah.
So about 10 hours ago.
Nice.
And we will continue diving until roughly 6:30 p.m. for us, so another little over 12 hours.
Kind of looks grumpy.
Oh, he does.
Almost like they just woke up and there's a giant yellow bus with lasers looking at them.
Almost like that.
So you mean excited?
Yes, like excited.
Video
Like
full
us.
wide.
I feel like all those things that feed on the benthos with the jaw on the bottom look grumpy, you know? Or more mopey, I guess.
Comes with the territory.
Yeah.
They're under a lot of pressure.
Hey-o.
Thank you.
Thank you for appreciating.
A double thank you.
One of the other cool things I know for me, as someone that doesn't like, and even my agency, we don't often get out to come to these places, is that things like this, like presenting it live and having scientists available to chat with and answer questions is just like an amazing opportunity, not only for classrooms, but just to like bring the public out to these spaces that it's nearly impossible to get to. So it's just a super cool opportunity.
Oh, crinoid.
What do we got? Is that
Looks like a crinoid. Yeah. But yeah, that's a really good point that those of us that come out as scientists sometimes think of it more heavily from the scientific perspective, but I think from the educational perspective, this is also incredibly important and beneficial, and just a different way essentially to teach about biodiversity and career paths and all these different subjects that wasn't always around for some of us when we were younger.
Yeah. Wow.
Yeah, it's beautiful.
These are so beautiful.
Quick, how many legs?
1, 2, 3, 4. I think like 21.
Wow.
21, 22. That's great. Thanks.
Is the number of legs, is that determined by species,
more
or
flat.
I think it's
will it depend on the
I think it's a factor.
Okay.
So especially for ones that are like 5-armed, I know that it can be like diagnostic, but I don't know if all species have like a designated number that they should have.
To kind of build on the idea of exposure to kind of cool careers, I know
Hey
that
Ben, can you zoom out on HiPAC for us?
as a teacher myself, like, you know, I don't think students are aware of just how many careers can bring you out onto a research vessel like this. So it really is.
We're not gonna.
Marley, we're a little hot.
I'm sorry.
It really is like a important chance for students and for teachers to see that it really does take all types to make something like this happen.
And Brandon, your role out on the ship as a science communicator is actually super fascinating as well, because you guys are working with classrooms across the country.
Yeah, we get to you know, beyond just this the dives, we also dial into classrooms. You can request to have people like us
thinking about
call in and kind of and speak to your students if that's the kind of thing you're interested in.
I got to watch Brandon and Holly do an interaction with a school in Honolulu, Holly, or?
Yes, they are in Honolulu.
Full wide video.
Shout out to St. Andrew's School in Honolulu.
You guys did a great job. That was really cool.
They changed in the plan?
I think so.
So if you're watching your teacher, and you can go to the Nautilus Live website, and there's a request form to have people like us, you know, speak to your students during these expeditions.
Can you zoom back in a little bit, Ben? Thanks. So the conversation is that there might be there's a desire for some rocks on the base of this kind of slide area. They think this might be a landslide. So down.
The Scoopy.
Yeah. So like down where we were.
So you want us to
go
back like this ridge?
Like more where it's the steeper part. So like.
You mean off the ridge?
Yeah, the darkest part,
I see.
essentially.
No, I think up in the higher section.
Roger.
Up here? Okay.
Can you?
Yeah, do you want to show
So you want to go
Ben?
back to the ridge and get a rock from the
You
ridge?
want to go
Yeah,
off the
I think
ridge
so.
and see if there's falls. Oh, I see. This little like.
Right.
Okay,
So
got
that's going
this
down 200 meters because those are 100-meter grid lines. The
so
white
this,
ones are. Yeah. Right.
Yeah. So it's going to be 200 meters over vertical over 100 meter. So you want us to follow along the ridge? Okay.
Okay.
Got it.
So
just follow
if we
the
had
white.
like 045.
040, yeah. Like over here.
Yeah. Like that.
Or,
Okay. And get a rock.
So over the lip. Okay.
And we'll try and get a rock there.
Sure.
So one more.
Okay. Yeah.
Okay.
So we're just having some internal discussion about where we're headed. We are kind of on a feature where there might have been a landslide, which is something that some geologists are interested in rocks from that part of the feature. So we're working with our
Bridge nav?
navigator to figure
Can I have
out
a ship move bearing
which direction to
050
go,
and
if
20
it's
meters
plausible to
temperature?
kind of drop back down a little bit in depth and stuff like that. So that's why you hear just a little bit of chatter.
First step will just get us back in line with the back of the ship.
Back row noise,
Yeah.
but some of the things that we were seeing on screen during that time, there was that big, beautiful, like stocked crinoid. That was the sort of the
All right,
large
cool.
pink fan-looking creature, and then there was a really tall. Was that a glass sponge that had
Yeah.
a bunch of were those, I think, brittle stars and crinoids on it?
Yeah.
So speaking of working with scientists from all over the place, we're working with requests for samples and trying to see how, whenever we can and it makes sense, to try to collect those collections.
Yeah.
Yeah, it looks like there's a lot of glass sponges.
All right,
Yeah,
Jess,
and a
I'm
lot
gonna
that are
head over so that we can bring your heading around.
dying as well, which we've seen on all the dives, really. You see that kind of like light brownish, yellowish color on the sponge, and that generally, for these types of sponges, is indicative that they've died. Some of that might be from sedimentation or just from age. I'm not really sure.
Yeah. How old do these sponges get?
I don't know.
We don't know.
Sorry. Someone might know, but I don't know.
And if it is steep, we'll end up moving Argus a little bit farther off of the cliff. So
Yeah.
Argus will be in deep water.
So as we're exploring, we've got, you know, Hercules, which is the main ROV that is down there with the primary camera that we're all often seeing, and then sort of back a little bit is Argus, which is sort of following behind. And so there's always an impressive level of coordination between the Argus pilot, which is sort of watching Hercules, which is way up close and doing the manipulations, and then both of them tethered to the ship, which all have to sort of move in concert. So there's always just this, just an amazing sort of dance between the two ROV pilots, the navigator here in the van, and then the ship itself.
Yeah, you guys are really good at your jobs.
It's really amazing to watch.
Yeah.
Ooh. If I go in a straight line right towards this target, it's going to keep us a little off the slope. Like, do you want me to go north and then, yeah, or do you want to keep us a little off the slope?
I would keep us off the slope more.
Good. Good, good.
So while they're still sort of like trying to navigate around to where we're going to go, the area that we're diving in right now, so we're in the Howland unit of the National Wildlife Refuge System. So Howland Island is a National Wildlife Refuge. It was designated in 1974 to protect seabirds on a small island.
We were pretty dense on this part,
but it's also
too.
located within the Pacific Remote Islands Marine National Monuments, which is one of the largest protected marine spaces on the planet. And that's managed by the U.S. Fish and Wildlife Service in partnership with NOAA to manage these spaces. And it's really reflects sort of a changing understanding of how like terrestrial environments and the marine environments are tied together and work together, and it's really important to be conserving both of them. And so dives like this really give us a baseline level of data to know what we're preserving, what we're caring for out here, and what's down here below the waves.
Yeah, we're just under 2,000 meters right now, so 1,993 meters. Nice, beautiful glass sponge.
Oh, that is pretty. It's like a trumpet.
Yeah.
Wow.
Or like a vase. Sometimes they're called vase sponges.
Yeah.
All right, Marley, go ahead and push it in there, please.
That is so pretty. Oh, look at all the fluting around the outside.
Yeah, how do those exterior projections contribute? You know, the filter feeding mechanisms on the interior.
Yeah.
That's weird.
Oh, really?
Yeah, that big cavity that it is, is this like just filter feeding matrix.
It's so intricate.
Contribution to beauty.
You're going to come a little wide there, Marley, and then we can get it all in shot there. That's good there.
Yeah, it's beautiful.
That's huge, too.
Yeah.
That's the size. Size large.
Size large.
All right, Marley, go ahead and come full wide, please.
Symmetry in nature. So cool.
Yeah.
Wow. We're seeing a lot more of those spiky sponges than we are of any more of those vase sponges. Is that your assessment of it, Alexis?
Yeah, there's definitely a number of those down here. I don't really know a lot about the those ones that are just have those long projections, but they must be pretty efficient in this habitat.
Yeah. I'm coming up a bit.
It's a little halosaur fish that we passed. And so we'll be diving. This dive has about 12 hours left in bottom time, and then we'll have a pretty extensive transit. We will be heading up to Johnston Atoll after this. Which, do you know the latitude for that?
No, I do not know the latitude of that.
We can look
we
it
could
up.
probably find that
We
up
have an
pretty quickly.
internet.
It's a ways.
We could ask our navigator.
Hey, Nav. No, I can look it up.
I know that we're
really
at the
zoomed in
westernmost
right now.
point of where we're going to be on this whole cruise.
Yeah.
But I don't know the transit time and
You haven't memorized the map of the Pacific?
you know, sometimes I like to live on the edge day to day.
Sometimes he likes to sleep.
Sorry.
Why aren't you a robot?
Johnston Atoll is
Yeah, but
at
we'll be mapping all the way there, so.
16.7295 degrees north and 169.
When we do that, those long transit mappings, we try to look at what has been mapped.
Yeah,
in
and
that
we're
area
currently at
and
like
try to match
1.6
to kind of complement
latitude.
the existing higher resolution
So,
data.
going up quite a bit.
Yes, I would love to do that.
Indeed.
So it'll be about 3 days, a
are
3-day
you off auto?
transit.
Yeah, I'm pretty sure.
We
there somewhere.
recently crossed the equator, which when you're aboard a ship and you cross the equator, there is a ceremony that happens for people that have never
Right.
crossed before, so that they may become shellbacks.
Join us.
Join us on the. So some of us who have crossed before will initiate the new shellbacks. They are currently polywogs. Will become shellbacks.
Several polywogs in the van today.
We have
How many are
quite a
there?
bit on board. So we'll have our ceremony after we start that transit, and then most likely be doing a little catch up on summaries of our previous dives, as well as there's a nice rat tail fish.
Yeah.
As well as maybe figuring other ways to entertain ourselves, like, I don't know, reading books, playing games,
Doing work.
doing work.
Processing all the data.
Oh yeah,
that
that's what I'm
we've been
supposed to say.
collecting 24 hours a day. for the
I was
last
supposed to say I'm
4
supposed to do my real job work.
Yeah. Johnston Atoll, also a unit of the National Wildlife Refuge
Yeah.
System. So it's a National Wildlife Refuge, also part of the Pacific Remote Islands Marine National Monument.
I brought us down a little. I don't want us to overshoot that.
Yeah, and so we'll have 3 dives up around the Johnston Atoll complex. So we'll dive on Johnston Atoll, but also on a couple seamounts in the area that have been only partially mapped or are unmapped. And then we have another long transit to Hawaii after that, which is where we will be ending our cruise.
And Ben, we're doing 045.
Yeah, I brought it.
Do you know
Thanks.
how long that transit is?
It's about 3.5.
Another 3.5.
Yeah.
So we got 3.5 and a dive sesh, and then 3.5.
Yeah. That's one of the
Vibes.
this dive sesh.
Dive sesh.
Can we take a look at that little bottle brush coral that's on the, it's just above your lasers to the left.
Sure. Oh,
there
good,
it is,
good,
yeah.
good
Top
eye
of the
there.
rock.
It's really cool. And some more like really cool of those branched glass sponges
Yeah.
that we've been seeing.
All right, video, go ahead and push on in there, please.
And a crinoid. Lots of things going on
Thank you.
on this rock.
A little pink something down there. I don't know what that is.
Let's get there, video.
Looks like a Chrysogorgia. There's a couple, at least one squat lobster in there. I don't know what that pink Oh, an enemy maybe at the bottom.
Yeah.
Now, is this named a bottle brush coral because it looks like a bottle brush, or because it works effectively as a bottle brush?
Because it looks like a bottle brush.
And who named it?
I mean, I don't know.
Sir Bottle Brush.
Clean freak.
Clean freak.
Sir, go ahead and come on your video.
Sir Bottle Brush of the Pacific Bottle Brushes.
We don't generally put our very precious coral specimens through cleaning training, so we don't know if it works effectively as a bottle brush.
I was just curious. You know, it wasn't because it didn't end up getting named rock candy coral, which I could
Also
see.
looks like rock candy.
Oh, I never thought
Oh, I
about
never
that.
thought about that.
Really brilliant purple down there in
Oh, the
little
Megan, you see the
cucumber.
transition change in Argus?
there's something
Yeah,
that's on that
definitely.
branch.
It's really sharp.
Just
What are you seeing?
there's the sponge on the bottom right, and then there's like something that's on a branch.
Oh, on the backside.
Yeah, could we
Sure.
just take a look at that?
Sure thing.
Thanks.
Crinoid?
I think so, yeah. Our video, go ahead
Oh
and push
yeah,
on there,
it
Marley.
does look like just a different crinoid.
4.
Yeah.
Oh no, this is
no,
not
coral.
a crinoid. Yeah.
Oh.
This looks like a type of black coral, maybe Umbellapathes.
I mean, it
It's
looks
like
just
a-
like a stock crinoid.
Yeah,
except
it
for-
did.
Wow. Are those polyps on the stalk as well?
Yeah, yeah. So some genuses have polyps all the way down the stalk, and some don't. That's great. Thank you.
Yeah, sure. Go ahead and come on, Marley.
Is there like an organization where the polyps on
coming
the
up.
colony for a branch would be working together in a way that was significantly together and different than like other branches? Or is it just each polyp is an individual part of the colony and there's no real significance of where it is on the branches that
Assuming sea
separate
spider.
them as
So
individuals
for most
or connect them?
For most corals, there's no difference. I have seen some black corals that have what people call sweeper tentacles. And like, so at the base, I've seen them at the base of a coral where it looks like the tentacles are really long. So like the polyp has really long tentacles, and is thought to like keep things away essentially. So like as a protective force, they're like a bit more, I don't know, stingy maybe because there would be more cnidocytes in those tentacles. But that's the only instance I can think of like real modifications in coral polyp structure.
I guess not just like the structure though. I mean like the whole thing is an organism, but are there kind of like sub parts of the organism itself that's functioning together kind of like in a significant way? But
You're thinking like siphonophores where you have like
Yeah.
some are the swimming
The swimming and
cells,
some are the
some are the
feeding.
feeding cells.
sure, yeah.
Because I don't think you consider a coral colonial in that same sense of differentiation functionally. But that's an
Yeah, no, yeah. It's generally like, so one, how do I say this? Yeah, so one colony, it's not one animal, but it is.
Right.
Like it's not one animal, but it all works together to make that one colony work.
Right.
And there's not differentiation between the polyps. For
Right,
the
you have
most
to
part.
conform.
This looks like a very
Individuality is ruled out.
tall but dead bamboo coral, except for that those top
Oh, the
bits
top.
of the branches might be
Yeah.
alive.
Marley, you want to push in a bit there?
What are some of the things that cause coral to die? Do they have a senescence, like a natural senescence, or is it normally environmental?
Look how big that is in Argus.
Yeah.
Probably. I mean, they have a natural senescence,
great,
I
Mariah.
believe.
Everything
but
does,
yeah,
But
Just stripped up here.
but I think most of what we see for coral death
Nice.
is just environmental. So, I mean, they're sessile organisms, so they settle in one place and they spend their whole life there. And they don't, there's thought that there's some chemical cues for where they settle, so they might be able to detect, like, oh, this is a potentially good spot, but they can't,
All right, Marley, go ahead and come wide there.
they can't know that when they settle. So sometimes it's just that it's not the best, there's not the best conditions where they started living, or it's good for a while, so it gets pretty big, but then over time, the conditions have changed, because these can live, some species can live for hundreds of years, some species can live for thousands of years. So to make it to thousands of years, you have to be in a place that has really good conditions for the majority of those
Current's
years.
picking up.
Yeah.
Well, and like for organisms that are living hundreds and thousands of years, just underlies the difficulty in
Stop blowing us
studying their
into the wall.
complete life cycle.
Oh, yeah,
right? 'Cause
yeah.
you're never able to follow one species
No.
for its entire lifespan. Or, I'm sorry, one individual.
Individual, yeah.
So here's a question on that giant bamboo coral that we just saw.
Yeah.
The polyps were growing on the top branches.
Mm-hmm.
Are they ever going to try and repopulate the lower branches, or are they just going to keep growing up?
That's a good question.
Yeah.
I don't fully know. I don't know the answer to that. I don't know if they'll grow back down. Yeah, I don't know. Sorry, Greg.
And this is why we science.
This
This is why we science.
And that's one of the things too, like, I mean, we come out here with some knowledge of what we are expecting to see and what we already know about an area, but I've learned very quickly to say I don't know and just be okay with that.
Yeah.
Because there's a lot of things that, like, we get asked questions, everybody has these thoughts about, oh, what about this? What about that? And so much, whoa.
Oh, ooh.
Whoa.
Oh,
Have a
that's
look.
cool.
Well done, Jess. Science is speechless.
Speechful.
Oh,
So we talk about finding a good spot to hang out.
yeah. This is clearly
This is
a good
cool.
spot.
All right, video, go ahead and push on in there, Marley.
Nice.
That is a healthy coral.
Yeah.
Looks like a number of different, maybe different bases as well, so it might be multiple colonies. Looks like a bamboo with lots of branching.
Yeah.
But it's branching right at the node.
Yeah, it looks like nodal branching, so that's where you see the branches coming off, where the little dark
Right. There's
patches
nodal
are.
and internodal.
Yes. What are
Pay
those
attention in
orange
class.
things on there? Oh, anemones maybe?
Oh, yeah, yeah, yeah. Do you have any more zoom in there, Marley?
Oh, yeah.
Yeah, those are anemones wrapped around.
Oh, really?
Yeah, so they will settle on coral, and then they just kind of, their disc will grow around, and then they'll feed there.
So we've got anemones, crinoids, brittle stars,
Squat
Squat
lobster.
squat lobster. Yeah.
There's probably some of those mouse shrimp.
Mice,
Mouse shrimp.
Nice. Sure.
mousy shrimp.
Oh, and there's a different coral. Oh, no, it's a
Yeah.
sponge.
Oh yeah,
it's
there's a sponge in the middle.
Yeah, it's like a whole big ecosystem.
Oh yeah.
It's one
Yeah.
thing.
So I like to call them neighborhoods.
Yeah.
Huh. Neighborhood corals.
Yeah, corals are the neighborhoods of the deep.
So this would tell us, one, that this is a good spot to be in terms of like the currents and
All right,
like
video, you and I come wide and we'll frame it up and then head out.
their ability to get, you know, food as filter feeders.
Yeah. This is kind of in a peculiar spot, but would this be possible to sample a branch?
Sure. I want to do one step back too if we have one dialed in. What might be a flying grab?
You might be able to land on the top of the rock, was my thought. We can do a team effort fly and grab if you want.
Either way, well, we can Try this out.
Rich, now can I have a
I think you just bother that shrimp, really.
220.
Oh, it's a
20
pycnogonid,
meters?
right?
Oh, center on the rock?
On the rock.
Probably. Want
Or
me
maybe
to
it's a
take a zoom
shrimp.
on it before I disturb it?
Yeah, let's just take a, if it's a shrimp, it'll move. If it's a pycnogonid, I don't know if it will. We might be able
moves if you
to
nudge
thrust it away, yeah.
Looks
Looks
like
like
a
a
pycnogonid.
Oh yeah, so a sea spider.
Oh, yeah.
Alright, you can come wide, Marley.
I have a feeling it's either very full or it's going to be very full after it finds that coral. They tend to stick their proboscis into cnidarians, so like anemones, or generally soft corals, and they will just kind of suck the life out of them for, yeah, I mean.
The sea spider one?
Yeah.
Oh my gosh, that's amazing.
Yeah. It's cool to see them with the, we find them with their proboscis in an anemone, and the anemone looks very withered, and the pycnogonid looks very pleased with itself.
Huh. Suppose I would be too if I had a big dinner.
Yeah.
Okay, let's try this out here.
Megan, where are we going to put this?
So is this like a quick kind of grab, or is a slurp a possibility?
Well, we are going to grab it, but it depends on how flexible it is as to whether we can slurp it.
Okay.
Do we have anything? Do we have anything open?
We have the 3 large bins open. So the forward, port side, and then the starboard, the 2 big ones.
It could also go in a small one that doesn't have a bamboo in it.
Sure.
If you want to double up.
Yeah, we can do that too.
Just because I don't want to put a coral in a big one and then we need to put a rock in there.
Yeah. We also have 3 slurps.
I wouldn't slurp this, just because it is.
Yeah, I figured it's a
pretty
little too
brittle.
straight.
Yeah.
Yeah.
Okay. Alrighty.
But we're looking for a piece that, if we can get a point where there's like a branching point, so just, there's a lot of it, so you guys can select what might look most suitable.
You want below the nose?
Sure.
Yeah, below the node.
Probably settle.
They're like continually losing.
Pleased,
Yeah. But ship and Argus are in good position, so you've got time.
Roger. Hey Ben, you want to do me a favor?
Yes.
Okay. Alright, that didn't work very well, did it? Alright, one more time.
Wow, just looking up pycnogonids a little bit, and there's over 1,300 known species of sea spiders.
Oh, wow.
Right. They're found in every ocean. So you find them in like Antarctica, really, really big. 70 centimeters, over 70 centimeters is the biggest one.
Dang,
What?
that's pretty crazy.
Yeah.
That's huge.
Alright Ben, one more time.
That looks stable enough.
Perched on top of a marble? Yeah. Okay, so note a slurp, and then we're putting this in the starboard box.
Yeah, let's go for the forwardmost little one, A.
Roger.
All right, I've got you on bubble.
Roger that. Oof, that's a good kick.
I know.
Try to stay stable. Okay. So anywhere you said is the
sorry. Yeah, we just want a branching point, but if we zoom in a little bit, there's a lot of different